Any lecture cannot be a success without the learned speaker. I, on behalf of INYAS and Amity Institute of Pharmacy, extend a very hearty thanks to the speaker of today's session, Mr. Vaibhav Kumar, who, despite his so much busy schedule, accepted our request and graced the occasion to share with us his words of wisdom and opinions. To Professor Chitme, who is Assistant Director, Amity Institute of Pharmacy, for his prompt response for approving this wonderful talk. I would like to take this opportunity to place on record our hearty thanks to Dr. Nishant Chakravarti, who leads INYAS as well as the Regenerative Medicine Lab at IIT Kharagpur for his perfect guidance and encouragement as he has extended to all of us. As students, you might be busy in running experiments, analyzing data, reading, writing, acquiring knowledge, and might be aware about developing new skills in yourself to make you future ready and get a suitable job role in pharmaceutical industry. But we rarely think about how to connect with someone to get that quality knowledge and guidance beyond our peers or teachers, especially the real examples that industry people come across with and deal with them to launch a product in the market. So idea of this expert lecture here is to first analyze what is the demand from the industry, what are they pursuing, how to meet the requirement of the industry. So this lecture is, of course, a kind of effort to give you that kind of exposure and knowledge directly from the industry expert. So let's learn in a more effective way towards updating our existing knowledge about the dissolution studies and acquiring new knowledge in this session. I hope this session will brew some interesting interactions in this direction, and that is why we are with an industry expert who has expertise with the dissolution concept, the advancements in that field, and thoroughly will navigate the IVIVC, emphasizing on the regulatory considerations. We shall have better insights to the biowaivers, which are the regulatory examples, and in some cases, especially for biowaivers, we get exemptions regarding certain drug products based on a quality evidence which demonstrates that bioequivalence is not at all required for in vivo bioequivalence type of studies. I mean, if we are doing it in vitro, so regulations will exempt such products to carry out the in vivo studies. In short, the topic of unraveling dissolution and its implications for IVIVC, the regulatory considerations, and biowaiver in pharmaceutical development is highly relevant for pharmaceutical knowledge seekers. It encompasses the critical aspects of drug formulation, optimization, regulatory compliance, and efficiency in the drug development process, ultimately impacting the quality, the safety, and availability of pharmaceutical products in the market. So, before actually starting the session, may I now request Dr. Chakravarti, Chair INYAS, to brief us about the origin of this initiative, its relevance, and a few INYAS activities. Dr. Nishant, please.
Yeah, thank you, Kalpana. Thanks a lot. You've introduced this topic, which is very relevant to everyone present here, very well. I thank on behalf of the organizing team and, of course, on behalf of the entire INYAS community, Mr. Vaibhav Kumar, for taking out of his busy schedule and being with us to give us an insight into.
Ananya, can you please mute others?
Yeah, that'll be great. Thank you so much. So as Dr. Kalpana has already mentioned very briefly about INYAS, but I would like to extend a bit more in detail so that most of the members who are present here have a better understanding of what our academy is and how it started and where we are right now. So INYAS stands for Indian National Young Academy of Science, and it is a very dynamic community of young scientists that was established by our parent organization, Indian National Science Academy. And it started in December 2014 with a vision to promote science education and foster networking amongst young scientists across the country. So since its inception, we have embarked on a wonderful journey so far, which has a lot of experience learning and multiple activities. I will just highlight a few of them in the next few minutes. We started with only 20 young scientists as founding members of INYAS. And presently, we have more than 110 members dedicated for its cause. The community is elected very competitively. So we have less than 10% chance of success with the applications that we receive very well. Wonderful applications that we receive across disciplines, including physical science, chemical science, biological sciences, medical science, veterinary sciences, agricultural sciences, earth and atmospheric sciences, and engineering sciences, mathematical sciences as well. So there's a diverse spread of disciplines that we are looking into. And now we are actually promoting science communicators or industry experts as well to become a part of our community. So in fact, in this last GBM, which was the 9th GBM for us, the general body meeting that was held in February 2024, we welcomed 30 new members from diverse scientific backgrounds, which has enhanced our academy's talent pool and expanding its outreach. So notably, we have maintained a very strong commitment to gender balance as well. So this time we have inducted 11 female and 19 male members selected across the country, which reflects our inclusive and diverse ethos. INYAS's activities have been encompassing a wide range of initiatives in science outreach, communication, training, and collaborations. So we are organizing a lot of workshops, webinars, scientific meetings, training programs for young scientists. In addition to what we are doing as outreach activities like science awareness camps, remote area lecture series, collaborations with our local communities, non-government organizations promoting science literacy and inclusivity. We do a lot of our work presently in an in-person physical mode, but of course, we do take the help of our online modes as well to reach out to the much wider community at large when we can do it, as is today's one of the meetings on an online mode. So some of our flagship events include Prayojan, which is like an event for PhD and post-PhD students, postdoctoral candidates to understand more about their opportunities. In fact, the registrations for Prayojan 2024 are happening right now. We also do a lot of activities for rural area teachers through RU Setup event. And then we also have a lot of activities for women scientists under the Wisdom series that we conduct. We also do a lot of competitions. For example, there is a competition called Saransh, the thesis presentation competition for PhD students. Then there is another thesis-related competition, which is known as the National Competition for Research Excellence. We also host this SciArt image competition, again for research scholars. Previously, in collaboration with IIT Hyderabad, we hosted this Model G20 initiative. We also do a lot of school-level competitions in physical mode, recognizing multiple talents across the country. So our impacts are spread across the borders as well now through our collaborations with young academies worldwide. So facilitating global scientific dialogue and cooperation. We are actively participating in science policy decisions and discussions across our governmental organizations as well. In recent years, we have achieved multiple milestones. We have secured international funding as well for our work. For example, the Inter-Academy Partnership has funded us for 2 projects, one of which is being led by Dr. Kalpana as well. Our endeavors focus on crucial topics, which include like ethics in scientific publications, awareness about predatory journals, science diplomacy, and more related to these topics, highlighting our commitment to addressing contemporary scientific challenges and scientific challenges in the global scale as well. So as celebrating INYAS's journey and achievements, it remains dedicated to its core values of the 4 I's, which include inculcate, illuminate, integrate, and innovate. Our future endeavors will aim to further amplify our impact, especially in the rural and remote areas, contributing positively to our societal development and advancing to the frontiers of science, technology, and innovation. We express gratitude to our parent organization, Indian National Science Academy, for its support, and we acknowledge the efforts by all our members, our core committee, and partners driving this mission forward. So, with a vision to continue growth, excellence, and societal impact, INYAS is poised to lead the way in science communication, outreach, and collaborations for betterment of our society and the scientific community at large. We are very fortunate in today because as we are celebrating the 10th year of INYAS's inception. So this year is a lot of activities being planned to commemorate this prestigious time that we have with us. And I think every step that we are taking and every event that we are conducting are in line with that. With this, I would like to thank you once again, Mr. Vaibhav Kumar, for your time and commitment. And I welcome all the guests and participants who have registered for this program and have given us their time. I hope this is a very fruitful initiative from our end and you get to learn a lot. Thank you very much, and over to you, Dr. Kalpana.
Thank you so much, Nishant, for enlightening the audiences about the relevance of today's session and making everyone aware about INYAS initiatives. Now, may I request one of our students of M.Pharm Pharmaceutics, Ms. Prachi, to kindly introduce the speaker to our audiences. Prachi, please. Okay. Ananya, can you please make her one of the host or so that she can speak? I think she's not able to speak. Please give her the rights. Prachi. Ananya, are you there? Okay, Prachi, can you come forward? She is in my class only. Speak.
We are not able to hear you, Prachi. We are not able to hear you. Kalpana, are you there?
May I request Mr. Vaibhav Kumar to please share his words of wisdom? So we all are eager to listen to you.
So,
Actually, there was a technical glitch at the start.
yeah,
So
we-
also I have some
Kalpana, please
announcements to
hold on.
make. One, the e-certificate will be provided to all the active participants upon registration for this event, whose link has been shared with all of you. So please make sure that you have entered the correct spelling of your name
Uh, Thank you, Dr. Nishant and Dr. Kalpana. Am I audible?
Yes, yes, I can hear you. I can hear you.
Actually, I connected from my mobile also, so that I do not have access through mobile to speak.
Okay, but we can hear you. Can you hear me? Hello?
Can you hear us, Vaibhav sir?
I can hear you, Kalpana.
Yeah.
Kalpana, Mr. Vaibhav's introduction was not audible to anyone.
Oh, okay.
Yeah.
So let me introduce everyone with the introduction. Yeah. So Mr. Vaibhav Kumar has a rich experience of teaching as well as industrial in the lead role especially. And he has total industrial experience of more than 17 years. He has worked with Sun Pharmaceutical Industries Limited, erstwhile Ranbaxy Laboratories Limited, from January 2007 to October 2020. And presently, he is an associate director, analytical research in Mylan Laboratories, Hyderabad. And his expertise is in risk assessment, method development, method validations, and transfer of analytical method for determination of trace level impurities like nitrosamine is his specialization in drug substances as well as drug products. It's our pleasure to have Mr. Vaibhav Kumar here with us for his lecture. So we are eager to hear to you. Can you please share your screen and come forward? Vaibhav sir, can you hear us? Sir, you are muted.
Hello?
Now you are unmuted. Yeah.
Am I
Yeah.
audible?
Yes, sir, you
Yes.
are audible.
Yes.
Okay, thank you. Thank you, Dr. Kalpana, and thank you, Dr. Nishant. So there is some technical issue with my laptop, I think. So I'll be sharing my screen.
Yes, sir, it is visible to us.
Yeah, screen is visible.
Yes, sir. Yes, it is visible.
Okay, fine. Thank you. Uh, thank you, Dr. Kalpana, to give me opportunity to speak
Pleasure,
about
sir.
uh, dissolution. So, regarding the selection of this topic, it was selected based on the request from Dr. Kalpana. Though I have a limited experience during my work in industry. Initially, I worked on dissolution techniques for about 6 to 8 years. After that, I have been taking care of LCMS, GCMS technique. So for this technique, I will try to cover the basics of dissolution and what is the importance of dissolution, what is in vitro, in vivo correlation, and what are the few regulatory requirements, and how we can compare our test product with the reference product. So in this presentation, we'll cover about basic introduction of dissolution. What are the various factors which can affect the dissolution rate? Few VCS classification, in vitro, in vivo correlation, how we set up the dissolution specification, and how we compare the dissolution profile of two formulations, illustrating with the case study, and what are the biowaivers. So first we can go through what is dissolution. Since we are taking the tablets or capsules in the solid oral dosage form. So first drug has to release from the dosage form inside our body, and then it has to pass various barriers and absorb into the bloodstream to reach the site of action to produce the action. So dissolution is a test used by the pharmaceutical industry throughout the life cycle of a pharmaceutical product, which helps to evaluate the rate of release of the drug substance from the dosage form, and also to maintain the batch consistency, how we are making the batches, are they consistent with the release profile or not, to establish the stability of the formulation, where during stability studies, what is happening to the drug product, is dissolution profile is out of range, or it is still in correlation with the initial formulation, and various defects from manufacturing of the drug products can also be tested. For oral solid dosage forms, usually two types of apparatuses are used, basket or paddle, and for immediate, modified and extended release formulation, usually paddle assembly has been used. And apart from these two, USP3, USP4, USP5, USP7, these dissolution apparatuses can be used based on the type of formulations which we are using. So in case of oral solid dosage forms, basket or paddle, two types of apparatuses are being used. We will cover here just oral solid dosage forms. As an in vitro dissolution test for the immediate release, solid oral dosage forms are used to assess lot-to-lot quality of the drug product, guide development of new formulation, and ensure continue product quality performance after certain changes, like when we are formulating a formulation and when we are going to scale up into the manufacturing location, there will be slight change in the manufacturing process, there will be slight change of manufacturing, and there is the change in the lot size of the tablet. So if we are doing scale up for the manufacturing process, we have to still compare of the product quality with respect to dissolution test with the initial formulation which we developed in the R&D. So this knowledge should also be ensured continued equivalence of the product throughout the product life cycle, as well as product sameness after certain post-approval changes also. Sometimes after filing, regulatory filing, some formulation has to be changed based on the availability of the raw materials, based on the availability of certain excipients. We have to change, sometimes have to change the source of excipient. So with that slight change, we have to file the changes to the agencies for approval, and after getting approval, we have to demonstrate that after this slight change in the product, still bioequivalence is not affected. So the bioequivalence has to be demonstrated first in vitro with the dissolution test. Drug absorption from the solid dosage form after oral administration depends on the release of drug substance, the dissolution or solubilization of the drug under our physiological condition, and the permeation across the GI tract. The drug formulation can go by deaggregation into the solution, by splitting into the fine particles, and finally go into the solution. Anyway, drug can go into the solution. After going into the solution only, the drug will be solubilized, and then after solubilization, drug can permeate through the GI membrane. So if we see what are the factors which can affect the dissolution rate, majorly three factors. One is formulation related factor, process related factor, how manufacturing process is being done, and product stability related factor. So in formulation related factors, there will be two components in drug formulation. One is API, that is active pharmaceutical, and others are excipients. So the property of API is very, very important for any kind of dissolution study. We have to understand the morphological form of the API. We have to understand the particle size, physical property, physicochemical properties of API. We have to understand the intrinsic property of API like solubility, hydrophobicity, what is the pK of the API, what is the solubility, what is the surface area of API, that we have to understand. Second component is excipient. In excipients, there are various kind of excipients used in the formulation: binder, lubricant, wetting agent, fillers, coating agent. So the type, the grade, and the particle size of these excipients also affect the dissolution rate. Sometimes the excipients can entrap the drug inside their matrix, so the release will be affected. So we have to have particular composition to achieve the desired dissolution rate by selecting the proper composition of the excipient. Next is the process related changes. So if how much the granulation, what type of granulation we are doing, dry granulation, wet granulation, how much blending time we are using, how much this will be the mixing time, how much the screen size of the mesh which we are using for granulation, and what about the temperature and time of drying we are selecting after granulation, how much compression force we are using for tableting. So if compression force will be more, then tablet will be harder and will take more time to disintegrate. The coating may also affect the dissolution. So these are the various factors related to the process of manufacturing of the formulation. And third one is the stability. So what is the proper stability condition where the tablet or capsule has been stored? So if it is stored at higher temperature, it may affect the dissolution rate. Higher humidity may affect the dissolution rate. Photo conditions, light conditions may also affect the dissolution rate. So these are the various factors which can affect the rate of dissolution, and we have to understand while formulating the formulation. We have to understand these all factors while establishing the formulation. Next comes to BCS classification. We all know that the drugs are majorly classified into 4 classes. First is class 1, that high solubility, high permeability. Class 2, low solubility, high permeability. Class 3, high solubility, low permeability. Class 4, low solubility, low permeability. So if solubility is higher, permeability higher, that will be classified into the class 1. So in this case, the solubility of drug is determined by dissolving the highest unit dose of the drug. Like we have various strength of the dosage form. That highest strength should be soluble in particular volume of the dissolution media at pH range of 1 to 8. So the drug substance is considered as highly soluble when the dose solubility volume of the solution is less than equal to 250 ml. High permeability drugs are generally those with an extent of absorption that is greater than 90% in the absence of documented instability in GI tract, or those whose permeability has been determined experimentally. The case 1, high solubility, high permeability drugs In some instances, high solubility, low permeability drug, that is 85% dissolution in 0.1 normal HCl in 15 minutes, can ensure that the availability of the drug is not limited by dissolution. Means dissolution here is not a rate limiting step. Only thing is, rate limiting step will be the permeability into the GI, from the GI tract to the bloodstream. In case of case 1 and case 3 drugs. In case of case 2 drugs, where low solubility is there and high permeability is there, then the drug dissolution may be the rate limiting step, because the solubility is low and a drug will be getting soluble with time. So that rate will be low compared to the class 1, case 1 and case 2, case 3. So if drug solubility is low, then dissolution rate of one formulation can be compared with the dissolution rate of another formulation. In that particular case, in vitro, in vivo correlation can be expected. What is in vitro? In vitro is dissolution, and what is in vitro? In vivo is bioequivalence study. So in vitro, in vivo correlation, we have to establish like first rate of dissolution of comparison of two formulations and rate of bioavailability comparison of two formulations, and then we have to compare both the formulation, both the outcome of the in vitro and in vivo studies to get the bioequivalence data. In case of case 4 drugs, where drug is very low soluble and very low permeable, in that particular case, oral drug delivery is not feasible. So in that case, dissolution will not be come into the picture. So as I said, in vivo, in vitro correlation is like it is being done in BCS class 2, where the value of dissolution as a quality control tool for predicting in vivo performance of the drug, which is significantly enhanced if an in vitro in vivo correlation is established. Acceptable products are bioequivalent in terms of in vivo performance, whereas unacceptable products are not bioequivalent. Bioequivalent to the formulation which is already filed, or bioequivalent to the innovator product. If we are working in a generic company, we have to have establishment of bioequivalent data of our generic product to the innovator product. So our generic product has to comply with the same bioequivalent study, with the same type of formulation, with the same dose, with that of the innovator product. As per FDA, to achieve an in vitro in vivo correlation, at least 3 batches that differ in the in vitro, in vivo as well as in vitro performance should be available. And if the batches show difference in vivo performance, then in vitro test condition can be modified to correspond with the in vivo data to achieve in vitro in vivo correlation. Sometimes, like while we are developing a formulation where dissolution data of our generic product is not comparable with the innovator product during bio studies. Since bio studies are very costly studies, a lot of cost is involved in the bio studies, and drug has to be administered to the healthy volunteers. Innovator as well as generic drug has to be administered under strict control to the healthy volunteers. That study is very costly. So we have to have some in vitro data to get confirmation that our generic product which we are manufacturing it may be comparable in vivo also based on the in vitro data. In simple words, what we do, the dissolution data of our generic product means in vitro data of our generic product should be comparable with the innovator product first. If that data is comparable, that will make sure that in vivo studies will also be comparable. In some instances, if in vivo studies will not be comparable, we can change our dissolution conditions. We can change in our in vitro test condition to modify the changes in the formulation so that we can establish proper in vitro in vivo correlation. Very often, the in vitro dissolution test is found to be more sensitive and discriminating than the normal in vitro test. From the quality assurance point of view, a more discriminative dissolution method is preferred because the test will indicate possible changes in the quality of the product before in vivo performance is affected. Means, what is the discriminative dissolution? Discriminative dissolution is the dissolution where it can discriminate between the bad batch and a good batch. Small change in the manufacturing step, small change in the manufacturing process, small change in the excipient source, small change in the storage condition of the formulation, that can be highlighted based on the dissolution. If that discrimination we can achieve, that will be preferred for in vitro dissolution test. Setting up the dissolution specification, there are 3 categories of dissolution test specification for immediate release drug products. Single point specification as a routine quality control test where we can establish some Q value, that is Q 80%. If Q 80%, that will be not less than 85% in 60 minutes, or less time we can specify like 80 Q in 30 minutes. 2-point specification as a routine quality control test for certain type of drug products where slow dissolving or poorly water soluble drugs such as carbamazepine, it may come under class 3 where solubility is low and permeability is high. So in that case, 2 time points can be established. One is 15 minutes to include the dissolution range, and at the later point of time, 30 minutes, 45 minutes or 60 minutes to ensure that the minimum 85% of dissolution is achieved. And third is we have to compare the dissolution profile, comparison of dissolution profile. So dissolution profile, what is the requirement of comparison of dissolution profile is first you have to compare with generic to innovator, and if our product is filed, we have to compare our initial product, filed product with the small changes in the manufacturing process or small changes in the excipient, means that is post-approval changes. To waive bioequivalence requirement for lower strength of a dosage form and to support waiver for other bioequivalence requirements. So this waiver I will discuss when waiver topic will come on the screen. So how we compare the dissolution profile? There are 2 factors. One is difference factor, one is similarity factor. Difference factor is called as F1. Similarity factor is called as F2. So this is the formula given for F1, in which RT is the dissolution value of the reference before change of our product, or T is the dissolution value of the test. Test may be post-approval changes also can be considered as test. Similarity factor is F2. F2 is this formula. So if we go with this formula, it will be not very easy because some lobe has to be determined. So there are various Excel sheets which is validated for F1 and F2 are available online. So we can open that validated Excel sheet and put our values for getting the F1 and F2 values. Dissolution profile comparison. What are the how we compare? A specific procedure to determine the difference and similarity factor. Determine the dissolution profile of two products, 12 units each of the test and reference. So we have to have 12 units dissolution of our product, test product, and 12 units dissolution of innovator product or our test product after before changes. So this using the mean dissolution values from both curves at each time interval, we have to calculate F1 and F2. And for the curves to be considered similar, F1 value should be close to 0 and F2 value should be close to 100. If the value close to 100 and F1 value close to 0, that those dissolution profiles are similar. Generally, the limit of F1 will be 0 to 15. If it goes to 15, it will be less similar. If it goes to 0, it will be more similar. But the limit is 0 to 15. So if the value is coming between 0 to 15, the profile will be considered as similar. And F2 value has a limit of 50 to 100. If it is close to 100, that will be similar. If it is close to 50, that will be dissimilar. So we better understand this comparison from this case study, in which the generic product can only be substituted with the branded version if they are bioequivalent with the innovator product. So when innovator product is already in market, so the generic products which are made in the same dosage form, in the same strength, using different manufacturing process, different list of excipients. So if generic product is formed, we have to match the bioequivalent data of our generic product with the innovator product. So for that, in vitro studies, dissolution studies will always be helpful. If in vitro data is comparable for our generic product with the innovator product, then there is a surety that it will be comparable in bio studies also. The product is considered as bioequivalent with the innovator brand when it contains identical amount of the same active ingredient in the same dosage form, and there is no difference in the availability at the site of drug action when they are administered at equal molar dose of similar condition. So here, if we see in active pharmaceutical ingredient of generic and innovator product is same. So means bio availability of API will be the same. Here the difference will be the difference in the formulation, and that difference in the formulation can be identified based on the in vitro dissolution studies. If in vitro dissolution studies is able to identify the difference between generic and innovator product, that will be helpful. So first we have to compare the dissolution profile of our generic product with the innovator product. If it is comparable, then we go for the bio studies. Bio studies has to be comparable, and then in vitro in vivo correlation has to be established. Bioavailability depends on drug dissolution and permeability across the GI tract. In vitro dissolution may be vital in assessing bioavailability. So in this case study, bioequivalence of 3 metronidazole brand was assessed in 3 different dissolution media by in vitro dissolution studies. So if we consider the sample A is innovator and sample B and C are our generic products. So we determine the hardness, weight variation, disintegration test, friability, and assay. So all these should be comparable first. If all these are comparable, then dissolution profile can be comparable. If we see here at pH 1.2, like 0.1 normal HCl, so all the profiles at 10, 15, 30, 45 and 60 minutes, all profiles are comparable to each other. At pH 4.5, all profiles are comparable with each other. And at pH 6.8, 10, 15, 30, 45, 60 minutes, all profiles are comparable, which we can see from F1 and F2 values. So F1 value for sample B is 4. That means it is more closer to 0. It is more similar to innovator product. Sample C is 5. It is little dissimilar, but since it is coming in the range from 0 to 15, it will be considered as similar to innovator product. Similar case of F2. It is 63, 64, 66, 58, 63, 66. All values are coming more than 50. So we can clearly see that our sample B and C are comparable to A across the different pH range from 1.2 to 6.8. So if our dissolution profile is comparable in such manner, it will assure that it will pass under bio studies also. Now comes Biowaiver. So in addition to the routine quality control test, comparative dissolution tests have been used to waive bioequivalence requirements. How we can waive the bioequivalence requirements that are considered as Biowaiver for the lower strength of the dosage form. Suppose a formulation has 3 different strengths, 25, 50, 100. So if 100 mg drug is comparable with the innovator product and there is no change in the formula composition, all batches are scale up, scale down, all batches are manufactured with the same blend, then the bio studies for 25 and 50 mg strength can be waived off based on the dissolution and bio data of 100 mg strength, based on the in vitro in vivo correlation data of 100 mg strength. 25 mg and 50 mg strength can be waived off. That will come under Biowaiver. For multiple strengths of IR product with linear kinetics, if immediate release formulation is there where kinetics is very linear, the bioequivalence study may be performed at highest strength and waiver of the in vivo studies may be granted on the lower strength based on an adequate dissolution data, provided that lower strengths are proportionately similar in composition. Thank you so much for listening. Thank you, Dr. Kalpana.
Thank you so much, sir. You have touched upon each and every aspect of dissolution studies, specifically with the start of it, like what dissolution is all about, the biowaivers, the regulatory aspect, and the case study, of course, the way you explained formulation A, B, and C, how the innovator one is there, and we are comparing the generic version of it. We have seen that formulation B and C were quite comparable across all the pH ranges that have been taken, like 1.2, 4.5, and 6.8. So I think with this perfectly, everyone would be able to understand both the aspect, similarity factor and difference factor, F1 and F2 concept altogether very well. So the session is now open for discussion and any questions if anyone have. So please raise your hand. Ananya, can you please check if there is any question in the chat box? or question answer session. Anyone among the audience? You can raise your hand. Yeah. Ananya, can you please make Sarvesh unmuted so that he can ask his question? Ananya? 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 Sarvesh, can you please write your question in the chat? Ananya? Ananya? Sir, just a moment. I am making him come here. He's in my classroom so that he can ask the question.
Sir. First of all, thank you for your wonderful session, sir. Sir, I just want to know that how can we validate our dissolution method, or what are the key parameters that we have to consider during the validation of a dissolution apparatus?
Your voice is low.
Hello, am I audible, sir?
Yes, yes.
So, first of all, sir, thank you for your wonderful session. And sir, my question is, how can I validate a dissolution method? And what are the key parameters that I have to consider during the validation of a dissolution apparatus?
Okay. So how you want to validate dissolution method, and how you can do that, and what are the parameters to be covered? Am I right? Okay. So if you are developing a method, any method, dissolution, assay, related substances, regarding validation, we have to follow ICH guidelines. And according to ICH guidelines, we have to cover all 9 parameters. Regarding 9 parameters, you can see ICH guidelines, where specificity, linearity, LOD, LOQ, method precision, accuracy, intermediate precision, robustness, solution stability. So these parameters have to be covered for every method, every analytical method.
Yes.
Welcome.
Any other question? Pragya. Sir, one more student, Pragya, will be asking the question. Good afternoon, sir.
Hi, good afternoon.
So if I want to release the generic product into the market, so should I consider the similarity factor within the range, or it would be better if it is near 100?
Sorry, I cannot get you. Please repeat.
There is your voice is actually echoing.
Actually, in voice, echo is happening.
Yes. Ananya, can you please help?
Ma'am, I can hear you. I guess there is some echo from sir side, Devo sir side.
Now you can speak.
Yeah, we can hear you, sir.
Go ahead. Yeah, please speak.
If I want to release a generic product into the market, so it would be better to consider the similarity factor within the range, or it would be near 100.
Ma'am, Web sir is not able to hear us. I guess there is some issue at his
Now it is coming clear.
Sir, actually, your voice is echoing. We are able to
subject, type your question in the chat box from the device in which you are sitting right now.
So I'll request all the participants to please write your questions in the chat box. Who has enabled the chat box option now? Please confirm whether you are able to
The participants can type their questions.
In the meantime, I request every participant, if possible at their end, and if turn on their camera so that we can take few pictures. To whomsoever end, if it is possible. That option is, I think, true. Ananya?
Yes, ma'am.
By the time we receive the
Uh,
question,
Grass.
are you able to do it now? Can you turn on your camera? No, not yet.
This Oxford also comes along, and Marshall Ganson to party because people campaign. Now, President Marshall was given. Marshall's talking about it. He's very interested. And the thing is, he's the best person when it comes to this. And because that's how it
Radha is also trying to write.
That's the professor wanted at Spain. Just
Trying to speak. Can you please write your question in the chat box?
already have
got the question. From saying good afternoon, sir. We want to release a generic product into the market. So should we consider the similarity factor of the product within the range of 50 to 100, or we can get approval only when it is near 100?
So Dr. Kalpana, your voice is echoing, but I can read the message from Pragya Singh. So that is the generic product into the market. If so, I consider the similarity factor of my product within the range of 50 to 100, or I get approval only when it is near to 100. So we cannot get approval based on this data only. We have to have the in vivo data in place also. So first, in vitro data has to be generated for our generic product. For that, we have to have comparison with the innovator. F1 value should be close to 0 and F2 value should be close to 100. So if it is coming in 50 to 100, that doesn't matter. Only matter is in vivo data should be comparable for the product. In vitro study being less costly to the industry, in vitro study is being done so that we are assured that it will not fail in vivo study. If product is comparable in vitro study, it will definitely be comparable in vivo study. I hope you get my answer.
Yes, sir, we got the answer. So just a request, if we have logged into 2 devices, can one of them be exited now if you don't want to share the screen? Maybe that is one of the reasons to get PC.
Sorry, Dr. Kalpana. Can you say again?
There are other chats also. Questions in the chat box.
Thank you.
We take them.
So while conducting in vivo in vitro study, there are several problems that result in not correlating with the value of in vitro and in vivo. And what are those factors and how can we reduce them? It's a very good question. So there are various, we know that drug dissolution is affected by various factors. So if in vivo condition, in vivo data is not found to be correlated with in vitro data, there could be some differences, some problems in the in vitro method, dissolution method. So we have to have validated dissolution method. If method is validated, then there should be optimized formulation process. So formulation process has to be optimized in terms where generic product is more comparable to the innovator product. Then only it will get in vivo studies part and in vivo in vitro correlation can be part.
Okay, we got the answer, sir. Anyone else have further questions? Any further questions? Okay, I think we are done with the question answer session. Thank you so much, sir, for this very well explained topic and accepting our request for the delivery of this wonderful session. Now, may I invite Dr. Nitin Sharma, sir, Head Division of Pharmaceutics, to formally deliver the vote of thanks. Nitin, sir. Okay, on behalf of him, respected Dr. Nishant Chakravarti, Chair INYAS, Dr. Chitme, and our most valued speaker, Mr. Vaibhav Sharma, all INYAS members and all the audiences who joined throughout for this particular session. I'm really happy and thankful to them. It's my privilege to have been asked to propose this vote of thanks on this occasion. And I also extend my thanks to AIP office, especially Mahesh Ji and INYAS office, Ananya, for their enormous cooperation as the technical support for this event. I also wish to thank our audiences for their patience, enthusiasm, and positive participation. Thank you once again to our learned speaker, Vaibhav sir. I think we can close this session if there are no more further questions. And all the participants who actively participated in the session, you will soon receive your e-certificates. Thank you so much.
Thank you, Dr. Kalpana.
Thank you, sir. Ananya, we can close the session. Thank you, sir. Thank you, everyone. Have a good day.